one of the very simple and interesting concepts that is actually disturbing every single medicos. Now, what is that concept? Coming to the point, beta 1, beta 2, beta 3, all these three receptors have got a very interesting receptor protein that is GS coupled. Means these all are actually GS coupled proteins, but the beta 2 of all these is acting just like the GI coupled receptor, but it is not GI, it is GS, yeah. B1, B2, B3. All are GS coupled receptors. Now, in today's lecture, let's crack this particular point in a very simple and easy way. First of all, when there is normal condition, okay, normal, what is seen? This particular receptor is actually stimulated by this drug or a ligand normally. This ligand, when binds to the receptor, this receptor gets stimulated. And we know GS receptors, when they are stimulated, they're responsible to activate adenylyl cyclase. And that is further responsible to convert ATP into cyclic AMP. Got. And now this cyclic AMP has got three important functions. Number one, to increase the calcium concentration inside the cell. Number two, this cyclic AMP is responsible to do the phosphorylation. Number three, this cyclic AMP is responsible to do to have certain sorts of actions on the channels present on this particular cell. Well, these three are the very important points regarding cyclic AMP you must remember. Coming to the next point, consider this as like two cells. Number one, cardiac cell. Number two, smooth muscle cells. Because I'm going to make people understand about the concept of the beta 2. So for that, I'm supposed to give you guys example of the two cells, cardiac cells and smooth cells. Smooth muscle cell, SM, and cardiac cell. Okay, this is the cardiac cell, that is smooth muscle cells. Coming to the point, first of all, normally, when it is about the activation of this receptor in the cardiac cells, in the cardiac muscles, okay? This particular ligand, when binds to the receptor, this is supposed to activate GS and AC. Then at the end, cyclic AMP is produced. In the very beginning, the very first step, we'll concentrate on the calcium. CAMP is responsible to increase the intracellular calcium, and this calcium has got a very important job. How like? Very simple. This calcium will come and will bind to the troponin present in between the actin and myosin. Now this particular structure is actually inactive structure. In this state, the cardiac myocyte is not going to have contraction. If the troponin is in between the actin and myosin, what if the calcium interacts with this troponin? Then, very simple, they will form a synthesis, they will do the synthesis of a complex. That is calcium and troponin complex. Now here is the complex synthesized. Now this complex is actually detaching this troponin from the actin myosin structure. What will happen now, very simple, is this troponin is removed, these actin and myosin, they are free to slide. So what will happen then is actin myosin slide, we know they will do the contraction. Got? Very simple concept. Coming to the next one. Now guess what? Again, this particular ligand is bound to the receptor and stimulating the GS coupled, and at the end it is again synthesizing what? Calcium. Now you're coming towards the smooth muscle. Now here in the smooth muscles, again this calcium will come and will find calmodulin, will interact with that calmodulin and will produce calmodulin complex. This complex is responsible to activate the myosin light chain kinase, which is actually an enzyme. So this enzyme will become activated. Now this is the activated myosin light chain kinase. Now this kinase enzyme is responsible to do the phosphorylation of the myosin light chains of the smooth muscles. Now these myosin light chains of the smooth muscles plus this inorganic phosphate, which is added by the myosin light chain kinase enzyme. Remember, myosin light chain kinase is enzyme which is supposed to interact with myosin light chains. Now what is the interaction mechanism of this enzyme? Very simple, it is going to add the inorganic phosphate here. Very simple. Myosin light chain kinase plus inorganic phosphate. When this myosin light chain is phosphorylated, it is then able to interact with the actin. Now what will happen? Very simple again. Actin myosin will interacts. What will happen then? Contraction. So here we got the concept of the cardiac myocyte and the smooth muscle cells. Okay. In the both the cases, there is contraction seen. As per our question, that is beta 1, 2, and 3, they all have got GS coupled, and we know about GS is a kind of stimulatory, is responsible to do the excitation of the cells, and here in regarding muscles point of view, it is supposed to do the contractions. Got? But the case is seen a bit different in the emergency condition. When we are in a stress state, we are breathing very fast, and the meanwhile the heart is pumping very fast. Heart rate is increased, and in the meanwhile again the breathing rate is also increased. Now we know this concept that a heart is covered with the beta 1 receptors predominantly. Okay, most of the receptors are actually beta 1. There are some other receptors available, but most are the beta 1. And regarding bronchioles point of view, lungs, in the bronchioles we have beta 2 predominantly available. Now what is happening here? If you want to breathe, so the bronchioles are supposed to dilate, and here we have beta 2. The mechanism of action of beta 2 is to do the contraction of the smooth muscles. Here the smooth muscles are not contracting in the emergency state, they are relaxing. The question is this, that why beta 2 is relaxing here, though it is a GS coupled receptor? And the answer is very simple. Just concentrate. Guess what? This is the beta 1, as well as it is actually, excuse me, this is the beta 1 receptor. Along with it, consider this one as beta 2 receptor. First of all, let's have a glance for the beta 1, how this is going to interact in the beta 1 perspective. The ligand binds to the receptor, activates GS, AC, ATP to cyclic AMP, calcium level increased, and it is going to interact with the troponin. Troponin detached, contraction seen. Heart rate is increased because beta 1 is available. Now coming towards the beta 2. Ligand binds to the receptor, receptor activated, move towards AC, again cyclic AMP increased, and this calcium is synthesized, calmodulin, contraction is seen. Now where is the relaxation point of view of this particular mechanism? Very simple, just concentrate. Now in the beginning I told you guys that here we have actually the cyclic AMP, which has got three important functions. Number one, to increase the intracellular calcium. Number two, to increase the phosphorylation. As this cyclic AMP is increased, these all will actually increase along with it. Okay, so in the stress condition, all the processes are increased. So what will happen is cyclic AMP is increasing the calcium level. In the meanwhile, it is increasing the phosphorylation also. For now, forget about the channels, okay? Because our concept is just about the beta 1, beta 2. Why their mechanism is different, though they are actually the GS coupled. Though they are actually the GS coupled receptors. Now this mic is dropping all the time. Coming to the point. Very simple, you guys are getting the point. So here we have now the beta 2 mechanism of action. Why beta 2 is different now, though here again the contraction is seen. Now, how this is going to relax the bronchioles? Coming to the point, the answer is here very clear. As this CAMP is increased in the stress condition, heart rate is going to increase. It's obvious, clear, easy. And in the meanwhile, this CAMP has got another job, that is to do the phosphorylation. Now, where will this phosphorylation will be done? And this is actually the answer of our question, which is going to differentiate the activity of beta 2 in the smooth muscles. How? Like, very simple. Now this CAMP, when it increases the phosphorylation, this phosphorylation will be on this myosin light chain kinase enzyme. This myosin light chain kinase enzyme, when this is phosphorylated, when the phosphate is added into this myosin light chain, it will then become inactive. It will be inhibited then. The case is simple now. If it is inhibited, then there is no any sort of interaction going to be seen with the calcium calmodulin. Means you can say in the short, there will be no any contraction at the end of the day. There will be only one thing seen, that is relaxation. So smooth muscles will relax. That is how in the stress, Actually, the receptors are stimulated, whether it is B1 or B2. B1 is supposed to increase cyclic AMP. Increase cyclic AMP will cause increase in the calcium concentration. That will increase the heart rate. And in the meanwhile, this increased cyclic AMP will also do the phosphorylation, and that phosphorylation will be of the myosin light chain kinase, and that is actually leading towards relaxation. This is why, because of which beta 1 on the heart is causing the contraction, beta 2 on the smooth muscles is causing relaxation, and though they all are BGS coupled. Hope you got the concept. Thank you for watching.